Hello to everyone. Thank you so much for joining us today. This is our inaugural webinar for the COMMeND group. If you don't know yet, COMMeND means Cross-disciplinary Otitis Media Mentoring Network Towards Diversity. So we plan to have webinars every 2 months. These will be recorded for those who cannot join us live and might be in different time zones. So we will have updated news, updated calendars posted on our website, which you can see in this slide down below. So just a few more words about COMMeND. This is funded by the National Institutes of Health of the United States, by the National Institute on Deafness and Other Communication Disorders. It is an R25, meaning it is a training grant. And our goal is to recruit and retain young investigators in otitis media research, thinking that it will be important for us to grow our group of investigators or researchers working in this field, especially those who are traditionally underrepresented in the sciences. So for year one, we gladly included 25 mentors who committed to mentoring our new mentees. For year one, we have 14 mentees, more than 20% of which are international, and more than around 60% are considered underrepresented minority individuals. For the activities aside from webinars, we also have one-on-one mentoring, focus group discussions, and they're also being encouraged to present their podium presentations in other international meetings. And so if you want to find out more, please go ahead and check our website. You can also join us or follow us on Instagram. All right, I'll stop share here. So today, I also want to introduce you to our MPIs first, because this is a multiple principal investigator grant. Aside from me, the other people who are running this program are Diego Preciado, that you can see here. He's now the Chief of Pediatric Otolaryngology in National Children's Hospital, and he will also be our second speaker for today. Maybe I'll mute and have Diego say hi to the audience.
Everyone, thank you for joining this first webinar for this COMMeND series. I'd like to commend, pardon the pun, Regie for her efforts in getting this grant funded for this program through the NIDCD. It was really her efforts and her labors that got us to where we are today. And I think it's a very exciting program and webinar series on top of all the other mentoring activities and associations with ARO and ISOM that this grant has facilitated. So great to see everyone. Regie, thank you very much again for your work. I mean, amazing. And thank you. Should I introduce Seweryn? You're going to introduce Regie, Seweryn?
It's your turn. I just wanted to mention also other members of the core group. That would be Alan Ryan, who's also MPI.
Absolutely.
The one who's been emailing you is Landon Mayer. You can show your video, Landon, if you like. And the other two would be Sonia Ziniel, our expert survey epidemiologist, and Rita Lee, who will be our last speaker speaking about collaborative leadership. Please go ahead and introduce Seweryn, and you can start sharing and start with the speaker series. Thank you.
Fantastic. So thank you again, Regie. It's really wonderful to get together and talk about otitis media in this forum, and it's fun to be able to lead these webinars, and we're very pleased and honored to be joined up just by you, Regie, but by Dr. Bialasiewicz, and that's Seweryn. I'm sure I didn't pronounce your name perfectly right, so I apologize.
Close.
But he's joining us from Australia. So he is at the University of Queensland in Australia at the Center for Ergonomics, at the Australian Center for Ecogenomics. He investigates the role of microbial ecology in health and disease, combining clinical microbiology with some emerging technologies, working on otitis media, chronic rhinosinusitis, pseudomonal infections, and cystic fibrosis, phage therapies, and other exciting innovations. So it's really a pleasure and an honor to have you in this group, Seweryn, and thank you for being the lead speaker for any of these series. This is the first series for this program, so I'm very much looking forward to your talk. And so with that, I'll turn it over to you, and I'll put my audio on mute. Thank you.
Well, Thank you for the introduction and the invitation. And from my perspective, good morning, everyone. Not sure where everyone else is. Good afternoon, good evening, all those good things. So with that, let's get going. I'll just share my screen. Okay. There we go. Now, everyone can see that? Good, perfect. Okay, so I'll be talking a little bit about just the update and general surface level snapshot about the microbiology in otitis media. But before I do that, I've been asked to briefly describe my journey to working with otitis media. So if you indulge me for a few moments, it's all because of these cute little fellas. And the reason why is that when I was growing up in Malta, I started watching all these documentaries, nature documentaries, particularly around about the environment from Australia. So that sort of started me down the journey of thinking about ecology. Then we moved to Colorado. So I did my undergrad in a university some of you may be familiar with. And I did a molecular biology degree, but also I just kept up that interest in ecology and added a environmental biology degree to that. So towards the end of my degree, I decided to do something about all those nature documentaries that I've been watching since I was a kid and go to Australia. And that's where I've been since. So in the intervening 20 some odd years, I was working for 17 of those in the largest children's hospital in Australia. And I was in a clinical microbiology research lab that was embedded in that hospital. So we worked quite closely with supporting the centralized pathology services and the clinicians at the hospital and around the state. And a lot of that focus was using molecular tools for diagnostics, for epidemiology, with a big focus on respiratory infections. So as time went on, I developed more and more of an interest in the microbiome aspect of it, because it still kind of touches on that ecology, because after all, we're just a series of different ecological niches, really. And from that, I ended up working more and more with the microbiome, and that led me to where I am now, about 5 years ago at the University of Queensland, at the Australian Centre for Ecogenomics. So essentially, in this center, we use a lot of different sequencing technology to look at the microbial ecology in different niches. My particular focus is in humans, but also it's extended to One Health areas, so animals as well. And I keep up that research interest in the upper respiratory tract. And with that came being able to tackle a particular issue in Australia, which is otitis media. And this is something that was brought to my attention many years ago through one of my collaborators, who is the ENT chair at the local hospital, Anders Seweryn. And so we started this quite fruitful collaboration that's still ongoing. And if some of you may be aware that in Australia, the First Nations community, the Aboriginal and Torres Strait Islanders, the problems with otitis media are quite significant, but I'm not really going to touch on that too much, because I believe someone else is going to be talking about that at a later point. So that's pretty much my journey to otitis media, but enough about me. Now let's talk a little bit about the microbes. So I wasn't sure how across the different methodologies everyone was. So I thought I'd just quickly outline some of the commonly used techniques. So the older studies tend to use traditional culturing techniques, and that's still used today. And they're great, but these techniques are not only labor intensive, but they're also very selective and don't really capture the broad diversity of organisms. So this is where modern molecular techniques have come in. First up is real-time PCR. So most of you will be familiar with that, considering the last few years of what we've gone through. The benefits there are they're fast, it's specific, it's very sensitive, and it can detect non-culturable organisms. But it can only detect a few of the selected organisms at any one time. So this is where 16S ribosomal RNA gene amplicon sequencing have come in, and it's been widely used to look at microbial ecology for the last 25 years. In essence, you just amplify a section of the 16S ribosomal RNA gene, which is a universally conserved gene across bacteria, fungi, all that sort of stuff. And it creates a fingerprint. And then you can categorize and classify those species, well, those bacteria. But the limitation is that oftentimes you can't get a good enough resolution down to species level. There's other considerations and issues there. I'm not going to really kind of go into that because that's going to be, I believe, covered later on this afternoon. I also wanted to mention metagenomic shotgun sequencing. There's haven't really been any studies looking at Otitis media microbiomes using this yet, but it's worth mentioning because it's something that it will be done at some point. And it's quite powerful because it sequences all the DNA in the sample. So you can imagine that any organism that has a DNA genome can potentially get captured and classified using this. And this can, it's quite powerful because it can potentially get you entire genomes, which will then let you be able to characterize the organisms down to species or strain level. But it does have its own drawbacks as well. And just some pictures of the two different technologies. This is a short read technology that sequences a lot of DNA all at once, and this is a nanopore, which is a long read technology. But you don't really need to know that at this point. So like a lot of other systems in the body, the upper respiratory tract is seated at birth. And there is a documented succession of colonization by different commensal bacteria, as well as some of the what's recognized as pathogenic bacteria later on. However, if there's disruption in this succession, particularly in the commensal colonization and early in life, this can then lead to an increased risk of future respiratory tract diseases, including otitis media. And what we're talking, so when we're talking about why that is, some of the ideas behind that, and these are still, people are still looking at this, is it could be during that first year of life, the mucosal immune system is still maturing. So it could be a maladaptation or a lack of established commensal bacteria being able to interfere with the pathogenic bacteria as they try to colonize the upper respiratory tract themselves. Now, in terms of the disruptions, it can be a variety of different things. It could potentially be early life use of antibiotics, exposure to the otopathogens, which are the three biggest ones are Streptococcus pneumonia, Haemophilus influenza, and Moraxella catarrhalis. In exposure to those in the first few months of life, certainly that's something that's been posited to be an issue with the First Nations children because they get otitis media quite early on. And that's potentially one of the reasons is that they get exposed to these otopathogens at a much earlier age than other children in urban settings. But the other one is respiratory virus infections. So I wanted to talk a little bit about that. Now, respiratory virus infections, they commonly precede cases of acute otitis media. And so the thought is that a lot of these bacterially derived acute otitis media are actually secondary bacterial infections. Common respiratory viruses that have been linked with development of ALM are RSV, human rhinovirus, influenza viruses, but that's not a a complete list. There's plenty of others, adenoviruses as well, and a variety of other cold viruses. In terms of mechanisms, there have been a few that have been proposed. So things like the virus infecting the epithelial cells, and then that in turn upregulating the otopathogens' ability for their twitch motility, which leads to increased adherence and motility. And so that then leads to more of an invasive phenotype. There's also been suggestions about a dysregulation of the innate immune adaptive immune systems. And so this then leads to the body's inability to clear the ingress of the blooming otopathogens that get into that middle ear. And of course, we need to mention SARS-CoV-2. There has been some work looking into this, but it's still early days and it's not quite conclusive. So some studies have found SARS-CoV-2 present in middle ear fluids, but there needs to be additional studies to really clarify if SARS-CoV-2 really has a role to play in otitis media. What it has, however, provided is an opportunity to gain some additional insights into the role of viruses in otitis media. So you'll recall in that first year of the pandemic, there was a lot of COVID-19 related restrictions. So that really, it's been well documented now that that's really caused a plummet in most of the different respiratory virus infections across the globe. And this has also correlated with a decrease of up to 78% in otitis media cases in children compared to pre-pandemic levels. So there's a suggested causal link between the virus drop and the subsequent decrease in rates of otitis media. But particularly interesting, I think, is that there's looking at the resolution of chronic OME. And so during that same lockdown period, OME, the chronic OME resolution has increased from 20% to 99% in that first year of the pandemic. So again, there's a suggestion that if you prevent recurring viral infections, you then reduce the re-triggering, I suppose, of the chronicity of OME and allow it to actually resolve itself. So this all talks about a disruption or dysbiosis of the upper respiratory tract microbiome being linked to development or ongoing OM. All right, so if we look at now, if we focus on bacterial community, we can then, by the way, you'd be surprised on how hard it is to actually find a decent cross-section of middle ear canals linking to the nose and the nasopharynx. So consequently, we've got creepy guy here. So looking at the healthy middle ear, most of the studies have found very low bacterial loads in this section. And some studies did find signals, but it's really quite difficult when you start looking and taking into account contamination. So if there is a native flora there, it is at very, very low loads. Now, if we look at the middle ear during AOM-OME, the most commonly detected bacteria using 16S sequencing is Haemophilus, Moraxellus, Staphylococcus, Streptococcus, and Aleococcus. Using PCR, which is more specific and we can get down to species levels, it's the usual suspects in middle ear fluids: Haemophilus influenza, Strep pneumo, and Moraxella catarrhalis. Once we get into the more severe end of the spectrum with otitis media with perforations, you start to see things like Pseudomonas, Olliogella, and it becomes, the more severe it is, the more likely it becomes a polymicrobial infection with secondary infections, possibly from the external ear coming in as well. Within the nasopharynx, there's been some work, and most of the recent work has shown that the microbiome within nasopharyngeal area isn't similar to that within the middle ear. However, viral detections tend to correlate in both sides, so that's a caveat there. Traditionally, adenoids have been thought to be the source of the bacterial overgrowth into the middle ear. However, recent 16S studies have disputed this and have found that the correlations between middle ear and the adenoid populations are actually different. Although, again, there are some other suggestions that maybe there's sampling issues and biases in that. So watch that space, whether or not that's actually connected or not. The alternative suggestion is that there is a disruption in the nasopharyngeal microbiome ecology, which of course includes the otopathogenic species, and that allows for those otopathogenic species to overgrow and then track their way up into the middle ear. Now, I wanted to just touch on about community and ecology. So, and this is just to illustrate the complexity of the interactions. And this is from a recent study that we did using 16S sequencing in some indigenous children here in Australia. And this shows a microbial correlation network. So essentially the darker and thicker the line, the stronger the correlation between the two bacterial genera. And so you can see here that the children who never experienced otitis media had a much more complex correlation network. And also here, if you can highlight this bit, is you can see that there's this genus Dulocigranulum. So there's one species in that, that's Dulocigranulum pigrum. And it's strongly correlated with Corynebacterium. And we know from our other analysis that this is Corynebacterium pseudodiphtherium. And that only occurs in the children with that never had otitis media. And the reason why I wanted to highlight this is because Dulocigranulum has been identified as a potential healthy, quote unquote, healthy bacterial commensal because of its ability to inhibit other pathogenic bacteria. So this is something that Catherine Lemon's lab has done. And they found that Dulocigranulum can inhibit Staph aureus by itself, but, and in line with what we were finding with our data, it needs Corynebacteria to be able to inhibit strep pneumoniae. And the mechanism of that isn't clear, but it's clearly dependent on those Corynebacteria species to be able to have that sort of behavior. So if we go back to these plots, we can see that pretty much in every category of child and their ear health, we see Streptococcus pneumoniae and Haemophilus influenza. So just the presence of these otopathogens isn't sufficient to cause disease. And what we found was that the difference, or one of the differences, lies in the amount of the bacteria in these children. So in each of these otopathogens, there were significantly lower loads in the children without any history of otitis media. So there seems to be a benefit in keeping the otopathogen loads low, especially in these young children. And this sort of then feeds into this idea that there's a protective commensal network at play, which may limit the overgrowth of these pathogens. And again, going back to this concept of having a more complex ecological network of different bacteria working together or perhaps against each other and keeping the potentially pathogenic bacteria in check. Now, I just wanted to finish up with comments on a couple of other things to consider. So otitis media is can be present differently depending on context of the population. So the worst clinical outcomes are driven by disadvantage. So there can be different rates of otitis media, different ages, different outcomes of the ear health, the disease severity, and this is really driven by disadvantage. So for example, First Nations communities, refugees, things like that. And so I just wanted to mention this because some of the studies that we see in the literature focus on more of the acute otitis media in relatively well-off socioeconomically urban centers. And so that can create a distortion and a sampling bias to what the microbiome will look like in these other more disadvantaged communities. So as an example, the strep pneumonia serotypes found in these urban-based studies could be very different from what you see in remote First Nations communities. So that's just a word of, I suppose, caution. And I believe later down the track, there will be some presentations dealing specifically in these topics. And finally, don't forget about all the other critters. It's not just about the bacteria or even the viruses. So just want to leave with this one last slide. So we did as an example, we did metagenomic shotgun sequencing. So remember, this is where we sequence all the DNA in the samples in this case, this is not an OM patient, this is a CRS patient. And we sampled sequentially, just from going straight from the front of the nose all the way back down through the throat. And what we found is in 3 of those sites, bacteria actually weren't the dominant species. In the vestibule, there was actually a fungus, and the other ones was a herpes virus. So, and all this sort of stuff works together in concert with the host immune factors as well. And with that, I want to thank you for your time. And this is apparently what the middle ear microbiome looks like according to the latest AI.
All right. Thank you so much, Seweryn. That was perfect. I will now proceed to the introduction for Diego because we will have our Q&A after the 3 talks, and hopefully we can also invite you to ask questions. All right. So as I said, Diego is one of our MPIs, and I'm so glad that he's co-leading this effort. As some of you know, he was the past president of the International Society for Otitis Media, and he's been in the otitis media research space for quite a while. Some of his studies have been seminal, I would say, especially those that focus on mucins and RNA. Those have been references for us here in the lab too. And so I won't blabber on because there's a lot in his CV. Let's just hear him talk about his take on clinical possibilities for application when looking at the middle ear microbiome. Thanks, Diego.
Thank you, Reggie. I'm going to share my screen. I loved your talk, Seweryn. There will be some overlap, and I think you make some really important points that we've learned from the ecology of otitis media. Some of which I think dispel certain long-held beliefs. And some that may guide innovation into the future. And I think the points of overlap that we'll have in our talks maybe have something to do with some of those concepts. So I will talk about the role of the microbiome in otitis media. I hope people can see my slides. I'm at Children's National Hospital in Washington, D.C., and it's really fun. And what a great pleasure to be talking to this group amongst friends that I see on TV here on Zoom, on the Zoom screens. So I'll be talking to you for about 15 or 20 minutes about these issues with some overlap on the talk you just heard, and maybe leaving you with some questions about the future at the end. I don't have any disclosures that are relevant to this lecture. I'll talk a little bit about the interface between the microbiome and the host immune system that Seweryn alluded to at the end of his talk. I do think otitis media suffers, as Seweryn said, from from mislabeling. Frequently, otitis media gets used as a term that is all-encompassing when we may be talking about a multitude of completely different diseases that could or may not be related in some continuum that has been discussed. And I do believe there is in some patients a continuum of disease, but not always. But if we consider the typical child with recurrent ear infections or the ensuing chronic otitis media with effusion, perhaps there is some degree of inflammatory continuum and what's interesting about the microbiome studies and other studies is that under normal conditions, the middle ear space does not have fluid, really, does not have a lot of secretory mucin production in normal condition, as opposed to the rest of the respiratory tract. It is not easy to access in healthy patients, so there's always proxies of the nasopharynx for healthy patients, and sometimes in cochlear implant or patients undergoing ear surgery for other reasons, you can proxy the middle ear space. But in a truly completely disease-free patient, it's hard to access the middle ear for sampling, either for cell culture or the determination of the microbiome or profiling in whatever you're trying to do. So most of the samples come from disease or some form of disease state. We do know that early inflammation occurs, that we have production of chronic mucins that are very typical or characteristic of chronic otitis media with effusion, at least. I think chronic otitis media with effusion is very different from chronic suppurative otitis media. It's a completely different disease process in some ways, which may or may not be related to each other. But we do have this concept of a continuum where you have early inflammation, maybe acute otitis media events, maybe some typical mucin features when you talk about mucins, and then you have this more chronic picture. And I'll get into that and its relationship to the microbiome in a little bit. We heard from Seweryn. There is the typical usual suspects in the middle ear of at least bacterial pathogens, with Haemophilus species being the most prevalent currently in acute otitis media and THI. Somewhat this has been clearly impacted by vaccination and pneumococcal vaccination, protocols that are pretty universal, that have shifted in some aspects the microbiology, if you will, of respiratory disease and certainly of otitis media. And I do think that over the course of the last 20 years, with the advent of pneumococcal vaccination, others on this call are way more expert on this than I am, but I do believe that in general, you can consider acute otitis media clinically having shifted somewhat in terms of the clinical phenotype of the disease from a disease that was more pneumococcal and in some cases more severe for some patients. You still have severe otitis media, and certainly COVID threw everything in flux, right? COVID kind of changed things, but the post-COVID era did change things. But in general, the invasive pneumococcus has perhaps become less prevalent in terms of the percentage of otitis media cases we see. And the majority of otitis media cases we see are more a Haemophilus picture. And I do believe the Haemophilus story correlates to the chronic middle ear effusion story a little more. I think there's a little bit more of a relationship there than with the pneumococcal clinical picture. And I'll show you some clinical data that may or may not support that. But we have more of a shift towards a mucin production, mucinous response than the invasive severe strep pneumo sort of picture. And that shift has occurred due to this vaccination. We've looked at the expression, mRNA profiling of mucins specific to the middle ear and the respiratory tract using cell lines, primarily from mouse or human or primary middle ear epithelial cells grown in culture. And what we have found is that after Haemophilus exposure or Haemophilus lysates, we see differential expression of the mucins, where the MUC5AC glycoprotein is expressed earlier and perhaps more robustly than MUC5B expression, which comes in a little later, at least in cells. And when you look at middle ear fluid, and you profile the mucins through unbiased approaches such as proteomics, which we did some time ago, we found that patients undergoing tympanostomy tube placement, and most of these patients are for chronic otitis media with effusion. That's the population that we mostly treat with tympanostomy tube at our hospital. You find that MUC5B is present in all middle ear fluid samples of chronic otitis media patients undergoing tympanostomy tube placement through proteomics, and that MUC5AC is captured only in a fewer number of patients, at least through proteomics and sampling all the protein content of middle ear fluid for patients undergoing tympanostomy tube. So it led us to make the statement that chronic otitis media fluid, and these are mostly chronic otitis media with effusion patients, MUC5B is predominant or more abundant or more identifiable or more prevalent, whatever you want to call it. And that's borne out when we've sampled or validated more effusions through Western blot, more specific assays for specific proteins, not sort of a global protein content approach, but you see Western blots, you always find MUC5B in middle ear fluid specimens pretty much across the board, and MUC5AC being more hit or miss in terms of its presence. So that's, I think, interesting. And when you go to the cells, you'll see MUC5AC being a more transient or early responder when you actually do bacterial protein exposure to middle ear cells. And what's interesting about the MUC5B story, and I'm just giving this as a preface to what the relationship is to the microbiome, is this is work from Colorado, primarily from Colorado, University of Colorado, that profiled the knockout mice now some 10 years ago. And I was so fascinated by their report or the characterization of the MUC5B knockout mouse, showing that these mice are susceptible to severe upper airway disease. And they made the determination that MUC5B in the mouse is required for airway defense, seeing infection, bacterial colonization, inflammation in the upper airways, ears, sinuses of mice, and decreased survival MUC5B mice. What the reasons for that? Not necessarily clear, but certainly correlation: increased infection, increased inflammation, and decreased survival. And well characterized, really well characterized mouse model showing the propensity to have fulminant otitis. Which is really interesting, right? Because what you see in the human is MUC5B is always present in middle ear fluid. However, if you take that component of the innate immune response away from a mouse, they're susceptible to severe infection. So clearly, MUC5B is a really important immune response to otitis media, acute otitis media, or otitis media pathogens. There is some relationship there that we see in mice and humans. Other proteins, you find in the middle ear include interleukin-8. That's a very classic and typical early response cytokine that responds to NTHI, to pneumococcus. And when you sample patient middle ear specimens, you'll find it in extreme abundance. There's also abundant DNA in middle ear fluid specimens, chronic otitis media mostly, that you find neutrophil elements, neutrophil extracellular traps. Many have shown this. Western Australia has really driven home the story of these neutrophil extracellular traps, which are associated again with biofilms, host component of biofilms, and you find biofilms in chronic otitis media. But certainly the fluid, if you just look at the fluid, you see waste of DNA, you see neutrophil extracellular traps, you see interleukin-8, you see MUC5B when you sample those fluid specimens. So this DNA that we had seen in our patient samples, we were very curious as to whether this DNA was mostly bacterial or host. We did some shotgun sequencing and found out most of the DNA was host derived, about 90%, with a small component coming from non-host. And certainly there is a correlation of the amount of DNA with the viscosity of the fluid. But we were very interested in terms of the non-host DNA, how what bacteria that DNA corresponded to, which led us to perform a microbiome study on 50 specimens and describe what we found in chronic otitis media fluid in those 50 patients. And we found the most abundant bacterial genre was indeed Haemophilus derived, and as you would expect in otitis media, but a host of other pathogens, which others have shown very similar sort of profile. And I really was enthralled by your comment, Seweryn, on how much the background, the ethnic and perhaps social background of these patients could influence this. We haven't looked at that, but I think that's a really fascinating comment. However, what we did do is correlate clinical profiles of the children that provided these samples in terms of the bacterial abundance and try to measure correlations there. What we found was that samples that tended to have more abundant Haemophilus were samples that had more mucin content as measured through the presence of both MUC5B and MUC5AC. So a really strong correlation of Haemophilus abundance with more mucin presence. We also found that age influenced the profile, at least in our patients, with older patients having a wholly different profile or microbiome profile, not necessarily Haemophilus related in the older patients, and the conductive hearing loss being more common in the Haemophilus patients. That is another way of thinking of more mucinous, perhaps more viscous story. So Haemophilus, mucins, conductive hearing loss, sort of those correlations exist, older patients being somewhat different. We looked at their respiratory history and found that patients with a lot of Haemophilus tended to have more asthma bronchiolitis histories as well. So correlation with the respiratory disease, and that sort of, like you just mentioned, Seweryn, more sort of single pathogen profile related to less diversity in their ecology. So the non-asthma, non-bronchiolitis being more diverse, as opposed to the more respiratory disease having more Haemophilus in their samples. So those are things we found. And so there are correlations between the microbiome and the clinical profiles. There's no question about that. And there are correlations between the microbiome and the host immunity. Which comes first, or what is causal here is unclear. Of course, these are just correlations, but there are clearly correlations that are telling us some form of story. Others have shown us, as to Seweryn's point, the idea of what's been profiled. This is a really nice summary paper, Seweryn, you're an author on it, led by Robin March from the Charles Darwin University in Australia, showing really what all microbiome papers have demonstrated, what species are typically found, and making some really interesting points that you alluded to. Are there some protective bacteria? What happens when you don't have diversity? Is there disease propensity when you have predominance of a pathogen? Why does that occur? And I think this is a really great summary paper. The paper from Western Australia that you mentioned, I think you mentioned, Seweryn, or maybe you mentioned something similar to what this paper found when they compared controls, microbiomes. These are healthy nasopharyngeal samples. That's what these controlled nasopharyngeal specimens are, versus patients with otitis media. You see a large number of patients that have really well done, N98 versus N86. They really start seeing some really interesting findings. The Dolocigranulum and Cornybacterium protection that you mentioned, Seweryn, was really noticed in this paper. So those patients that had those species or those bacterial genera identified in the nasopharynx not having otitis media. So there clearly are some relationships between bacteria that are not associated with disease, and some that are, right? And so what's happening, I think, is going to be important as we consider ways of preventing or treating otitis media. So some have looked at these probiotic or bacterially protective means of decreasing pathogens or decreasing pathogen load through competition or through, competition for nutrients. A probiotic in some measure, or a way of controlling the microbiome to make it less pathogenic. So some phase 2 trials looking at alpha hemolytic streptococci as a way of competing away for the strep pneumo that are currently ongoing. And the guidelines by and large still do not propose probiotic strategies as a recommended means of treating or preventing acute otitis media in children. But I think over the next 5 or 10 years, we're going to start learning more about this and hopefully finding some approach that will be useful or clinically relevant in most cases, or for some children. I do believe there is a path towards this type of approach. There's this paper also from Colorado, in collaboration with Dr. March and in Australia at the Northern Territories University there, and then investigators from, I believe, Tasmania. I really enjoyed this paper. This is pretty recent, but this is preclinical. This is done in mouse models and in cell lines, where they found pretty good evidence that approach of competition through non-pathogenic pre-exposure in the respiratory tract of mice, in this case they used Haemophilus hemolyticus, leads to decreased NTHI burden after challenge in both the nasopharynx and the lung, using different types of Haemophilus hemolyticus. Not only that, but they identified the protein that Haemophilus hemolyticus expresses or uses to compete off the nutrients, and this is hemophilin, which is a protein that takes away that iron source, the nutrient iron source for Haemophilus NTHI. It uses it up, and that's the mechanism through which these more commensal hemolyticus species seem to protect mice when treated either with recombinant hemophilin or with actual Haemophilus hemolyticus bacteria as a means of decreasing bacterial load in the respiratory tract. Really convincing, really well done study that I think informs potentially what future approaches will hopefully look like in patients to prevent NTHI infection. How we deliver these proteins or these bacteria, it's not necessarily clear, but you can start imagining means of controlling the microbiome to benefit and decrease disease burden. So in summary, I think the microbiome, as Seweryn mentioned, the microbiome studies do identify bacteria not previously described by typical culture studies, and that there is a correlation to immunity and host response that you can see in patients. As you said, Seweryn, the adenoid reservoir theory is not as clear because you see distinct profiles of middle ear and nasopharynx through microbiome studies. There are potentially protective bacteria, bacterial genera, so the Corynebacterium dolosigranulum being two that have been identified in the nasopharynx of healthy non-otitis children, and probiotic strategies, such as that example from the Colorado study using Haemophilus expressing bacteria to leach off iron nutrient sources may serve in the future, hopefully clinically, to some relevant fashion. So I appreciate this opportunity to talk, and of course glad to keep this discussion, as there's a lot of questions being raised through some of these descriptive studies, and then these preclinical studies. So these are some of the people that I had the real great honor of working with over the years that really are responsible for most of this data that at least came from our center. So thank you guys so much. I'll leave it at that, and I'll turn it over back to you, Regie. Thank you.
Thank you, Diego. Some really nice questions in the chat, so please hold off and just keep putting in your questions. In the next 10 to 15 minutes, I'll try to be quick. I'm sharing my screen again. So for those who just joined, this is already the third talk If you just wait a little bit, we'll be posting the recording on our website, and you can catch the first two lectures. You can also tell your friends who can't join us today. So for today, I thought that especially for the junior investigators, I just wanted to give you an idea of what are my learnings doing microbiome research for otitis media and some of the limitations that we had to encounter and go over to push through with some of our studies. At the same time, what would be the opportunities that come out of these learnings? Just to give you an idea how I started, I'm actually an otolaryngology trained person from the Philippines, from the Philippine General Hospital. And after my 4 years of residency, I thought I'd have a sabbatical and work on genetic epidemiology. I got a master's degree first from Erasmus Medical Center, Rotterdam, and then I got hooked and ended up doing a PhD. After my PhD in genetic epidemiology, I went home, started to practice there in my home university. And then Dr. Rina Reyes-Quintos invited us to a medical mission. And when we landed in the island where the indigenous population is, at that same night, we recognized that they were all intermarried, and we ended up over the years building this really nice pedigree. These missions that we've been going through in that population was led by our current dean in the University of Philippines College of Medicine, Charlotte Chong, who's also a neurologist. And so as of now, we've identified here you can only see 3 genes, SPINK5, A2ML1, and FUT2, but there's actually a 4th gene that we just submitted for publication based on the work of my previous PhD student, Christina Elling, who is also here. So we now consider this pedigree solved after how many years of linkage studies. But going back to the history, then why did I get involved in the microbiome? So, you know, as you can see in the right panel, we've replicated some of these findings for A2ML1 and FUT2. You can see here on the right all the different variants that have been identified in A2ML1 in the Finnish, United States, and Filipino general populations. So we know that this is a real finding, but then why go into the microbiome? So the story is, I was research assistant professor at Baylor. I belong to a different lab, and just so happened Professor John Belmont, he's a pediatric geneticist, now retired, an HHMI professor, well known in the field. He just so happened to be collaborating with the lab I was in, and he knew that I was working on this pedigree. At the time, we were just starting with foundation grants. And so one day I got in the elevator. It was a very short ride because it was, I believe, just 2 or 3 floors, 2 floors at Baylor, the main building. And as soon as I came in, he said hi and said, why don't you work on the microbiome? Just get some samples from your population and just submit them. So, you know, within a few seconds, that just changed my trajectory. Instead of just focusing on the genetic linkage, we ended up getting some microbiome samples together with Dean Chong and Dr. Rina. We got some samples from the population 2014, 2016, and 2018. And so that got the ball rolling. First, we got a foundation grant from the National Organization for Hearing Research, and then eventually an R01 that combines genetics and microbiome for otitis media. So, you know, lesson there, always look around when you're in the elevator, you might just get some really good ideas. So when I joined Colorado, University of Colorado in 2016, I started working with Dan Frank, who's an associate professor here in the Department of Medicine, Infectious Diseases section. And he's such a great collaborator to work with. It's because of his attention to detail, rigor, and patience with our studies that we're able to find some really great microbiome signals in our indigenous population, and also here in the children that have otitis media in Colorado. So here's an example of his results. We submitted some samples for the middle ear swabs. These would be community-based swabs that we get from the edges of the perforations of the eardrum in those who are affected with chronic otitis media and a very few recurrent acute otitis media. Then we also get swabs from the outer ear, the nasopharynx, and the oral cavity. So you can see here that based on one variant in the gene SPINK5, You can see different profiles depending on whether there's a variant in the patient or not. So you can also see here that we only have significance in the oral cavity for SPINK5 variants. And it's pretty clear that from the bacterial profiles, from the 16S profiles, those in the oral cavity would be different from the nasopharynx, although there is some similarity between the outer ear and middle ear. But then again, these would also be different from nasopharynx and oral. So very known fact that even if you use nasopharynx or oral cavity or adenoid as a surrogate for your middle ear profiles, it will always be different. So if you now look at the right panel, what we found was in the SPINK5 carriers, there's this nominal significance for Microbacteriaceae here encircled number 9 in the outer ear and the oral cavity, but then it's relatively significant in the middle ear. And then again, if you look at the nasopharynx, there's this 56 that is leptotrichia, which when Dan tried to replicate our original study for the A2ML1 microbiome, it turns out that leptotrichia is really significant in terms of increased abundance in the middle ear. And so we thought that that was a very significant finding. And again, I attribute that to the collective efforts of the team in the Philippines and also Dan Frank's attention to rigor with his studies. A more recent study that we published would be this microbiome study in the Colorado children with Down syndrome and otitis media. As you can see here, there is a very obvious difference between the bacterial profiles in the bar chart on the left. This is a beta diversity study wherein you see there's more diverse population of microbial communities in the Down syndrome kids versus those without Down syndrome, both having otitis media. And then if you look in the right, in terms of relative taxa abundance, you're seeing DE-seq2, you can see that there are more bacterial taxa that are involved significantly in Down syndrome versus that in those without Down syndrome. And this is work that was done by Christina Elling, who was also a postdoc for a few months with us, now an analyst with GeneDx. And so for my task today, so what are the challenges that we encountered over these years that we were working on the microbiome? So first challenge, the lack of normal controls. As you can see in our populations under study, we usually get middle ear samples from swabs, whether that's in the community or at surgery when a child is undergoing ear tube surgery. When they're inserting ear tubes, you cannot really sample the actual middle ear cavity, but instead you swab the middle ear, the tympanic membrane when the surgeon is putting in the tube. There are very few cases where we have open surgery for tympanoplasty or mastodectomy that you can swab that. And that's like 5% of the pediatric population undergoing surgery here. So we do not really have real controls in terms of swabbing the middle ear. And then the other thing is the Human Microbiome Project does not include middle ear samples. The GOSRA are biobanks for microbiome, and you only have very limited data available from affected individuals with middle ear samples. So again, most people use the nasopharyngeal swab as a surrogate, but then that's also a different niche compared to the middle ear. And so what we try to do, like other people, would be to collect swabs from children undergoing cochlear implantation, especially those that do not have a history of otitis media, but we ended up not getting anything. So our quality control pipeline says that if we do not have more than 10 nanogram per microliter concentration or more than 2,500 reads from sequencing, then we have to exclude that sample from analysis because it might end up being biased. Given that middle ear samples are usually low biomass samples, you can easily be swayed by something that's a contaminant. So we dropped this study, we closed this protocol because we were not getting anything. And so the question that is posed right now in the community is the healthy middle ear really sterile? And if so, then is there really a way that you can use healthy middle ears as a control if it is sterile, right? So that's the problem. It remains controversial. If you submit a paper or a grant application right now, a lot of reviewers would say you do not have real controls. You're only getting samples from affecteds. But then the evidence already from other studies show that whether you're getting swabs from cochlear implantation, from stapidotomy, or translabyrinthine approach for vestibular schwannomas, these are relatively clean surgeries, even with no history of otitis media or maybe something that's from far back. They usually do not get anything. And even this other study from 2019 from Jervis Bardi et al., they even sequenced the bacteria that they identified with sequencing, they submitted a report for gram stain and culture and then ended up not growing anything. So they say that this is a ketome, essentially a spurious signal for contamination from reagents that you use for sequencing. And something similar from Sarah Libera's group, if you have middle ear rinses during cochlear implantation, majority of those will end up being negative for any bacteria. And you might, if you push through with those samples, you might end up with high risk of bias for contaminants. And obviously your pool for controls will be very limited. So that's my first take. Challenge number 2, of course, the other thing that's limiting our sample sizes would be lack of access to middle ear. And so our cross-sectional data becomes very limited in terms of the sample size. When the nasopharynx and the adenoid or oral cavity are used as surrogates, as I said earlier, if you compare the bar charts, they're very different in terms of bacterial profiles. And that's the same when you're doing principal component analysis. You can see here there's some overlap with outer ear and middle ear, but the nasopharynx and oral cavity are very different. The other thing is, since we're working on genetic signals or genetic expression, some of our models might be unique to the middle ear. So we cannot really use the other sites as surrogate if we're specifically looking at a gene for replication. And so although large sample sizes are ideal, sometimes you also have to look at potential heterogeneity that might remove your signal if you're doing association analysis for a microbiome study. And this has led to false negative results, example, in large scale studies for gut microbiome. So what's the opportunity? What we've done is really look at extremes of phenotypes. That's a classic epidemiologic tactic that's used not just in general epidemiology, but also for genetic epidemiology. We've looked at very strong genetic variants. We've looked at extremes of age. So if you see this panel on the right, if you compare those that are more than 50 years old or 40 years old compared to the teenagers who have cholesteatoma, they have very different profiles. And in this case, age becomes a surrogate for duration of cholesteatoma or chronic suppurative otitis media. And so you have kind of like a mimic of a natural history if a cholesteatoma as part of acquired otitis media becomes not treated as well as when you are in a better health system. So a classic way also of looking at otitis media status would be comparing chronic suppurative otitis media versus a single episode of acute otitis media, which would be very different, obviously. There have been also studies on different types of chronic otitis media with diffusion, depending on what your fluid is, whether it's serous or mucinous. So there are several things that you can do for hypothesis-based analysis, whether it's other comorbidities like the lung, or in our case, we looked at Down syndrome. Though it's genetic, a lot of that is really immune related. And one thing that's really missing in the field would be longitudinal profiles. Hopefully, more people would be looking into multiple sampling from the same individuals. And then over time, how does the microbiome look like? Just another example of our microbiota data from cholesteatoma here. We looked at the differences if there's a cholesteatoma versus not, and obviously there are changes in the alpha diversity and beta diversity of the samples. And then again, if you put in antibiotics such as quinolones, there's also some changes in alpha diversity. You can see here that there is greater richness and evenness when you are doing microbiome study for antibiotics. And so challenge number 3. So most of our publications right now, this has already been mentioned by Seweryn, they are limited to 16S RNA gene sequencing and focusing on bacteria. usually only up to genus level. And Dan corrected me that it's not really because we're just doing 16S, it's because we're using short reads. And so we are sacrificing the amount of data that we get to go to species level by using short reads and making them deeper in terms of sequencing, having more copies that you can scaffold in order to determine the sequence for specific bacteria. But if we were doing long reads, then we can possibly do species level just with 16S sequencing and even phylogenetic analysis if somebody knows how to do it. Apparently, it's now a lost art because not too many people are doing that. The problem is if we only have genus level and not species level, bacteria from the same genus can have very different effects. So for example, strep pneum is a very well-known pathogen, but they're also looking at strep salivarius for potential probiotic. So these things we really have to find out at the species level. And obviously, if we don't do metagenomics, then we don't have as much information with viral, fungal, or archaea data. And there are only very few examples of bacterial, viral, fungal profiles in the literature. One classic study is from Tausishan Maitri, one of my best collaborators. She looked at a comparison for otitis media following upper respiratory tract infection due to viruses versus not having OM. And in this study, they showed that with 16S, Pseudomonas and Marshalla are increased in otitis media kids following a viral infection. So that's a nice longitudinal study that somehow combines bacteria and viral profiles. So again, there's a great opportunity for metagenomics because that has not been done yet for otitis media. There are a lot of studies on whole genome sequencing of isolates, but this is not community level data. There's one study from China wherein they combined bacterial fungal profiles and they found that Aspergillus is actually present in around 70% of Clostridioma, although their sample size is quite small. And then the occurrence of Aspergillus was associated with bone erosion in the CT scans. Challenge number 4. So I come from the multi-omics space. We look at genomics, transcriptome, and also microbiome. Our lab definitely does them now separately because we just haven't had the opportunity to have the same individuals with the sequence data for all of these omics together. And if we had that along with clinical data, that will be really great for multi-omic study, a real multi-omic study that if there's deep enough information, we can do machine learning. Here's an example of what Dan did with some of our data from the SPINK5 and FOOT2 microbiome. He used machine learning to see the differences in the microbial communities, just to show that, you know, this is doable. But unfortunately, there's not enough information for us yet to have this published. Challenge number 5, so this is also mentioned from the previous 2 talks, that the microbiome might be personalized or cohort specific, especially given the differences in the social cultural profiles of the cohort that you're looking at. In our case, we are concerned with gene-environment interactions because the families that we're looking at, they share both genes and microbiomes, and how to tease that apart might be a challenge sometimes. And then, of course, there are differences, as mentioned, in healthcare systems and access to otolaryngologists or pediatricians with a specialization in otitis media care. So some of these profiles, though, are also difficult to model in animals or cell lines because most of what you see in cell lines or animal studies, you do that bacteria by bacteria or virus by virus. You don't really put an entire microbiome in their ear. Just to mimic the human autopathogens. And especially in the mice, they have their own microbiome too. They don't really carry the human autopathogens. So that's a challenge. But I think there's also this opportunity that if multiple studies are done across different institutions, different cohorts, then we can replicate findings across different groups. As long as there's enough rigor in the study design, then we can replicate our findings. And I leave you with one one of the quotes for the ISOM Panel 4 that was published earlier this year. The panel includes Seweryn, but this was led by Ruth Thornton and Sharon Tamir, who are COMMeND mentors. They said that there's still some replication from their review of the microbiome studies from the past 5 years, with some degree in the overlap of the AOM or OME, different types of otitis media samples that they see in the literature. So there's likely a microbial continuum. And in that case, even though our cohorts are quite different in terms of background, then there might still be some overlaps in our findings. So, and then lastly, because of our multi-omics slant, we end up with hypotheses on potential mechanisms for otitis media pathogenesis. So in summary, there are plenty of opportunities for microbiome research in otitis media. And even though we might have studies from different cohorts and institutions, they might replicate rather than compete with each other. And alone or combined with our omics, microbiome studies can generate hypotheses on OM mechanisms, which are important if we want to translate our findings to clinical practice, whether it's to guide our antibiotic treatments or to make new therapies such as probiotics. All right, and here are acknowledgements. We really thank the Frank Lab, my peeps here, including Christina, and then our other surgeons or faculty, staff, CRCs here at Children's Hospital, Colorado, and UC Health. And of course, a very big group led by Charlotte Chong and Rina Reyes-Quintos from the Philippine General Hospital, with an army of residents helping out with collection of samples and recruitment of patients. I'll stop share. All right, so I think I went long. Maybe you can ask the first question now. So let's start with Stephen's question. Has there been any findings on human herpes viruses being able to enhance infection similar to respiratory viruses? I think this is a question for Seweryn.
Yeah, look, not that I'm aware of. The herpes viruses, they're a bit of a different beast to the standard respiratory viruses because they tend to become latent rather than just acute transitory infections in most respiratory viruses. So a lot of times, for example, in that patient, what we think was happening it wasn't a primary infection, but it was a reactivation, probably due to the inflammatory response from his CRS. What that means in terms of the microbial shifts, we don't know. Certainly, there's been some work done in, there's been correlations with herpes viruses being correlated with worse clinical outcomes with lower respiratory tract infections. But again, is it a cause or effect? It's not clear. My personal feeling would be probably it's more likely that the reactivation of herpes viruses is more of a indication of pre-existing issues and inflammatory responses from a bacterial infection, for example.
So just a follow-up question. Did you find the HHV in the middle ear or in other parts of? I think you showed us the slide.
Yeah, so where we found it was in the middle meatus and in the nasopharynx.
Okay.
So yeah, and that's because what we think is happening is that some of the herpes viruses tend to become latent in the neural pathways that go through the nose and into the sort of the oropharyngeal area. Yeah.
Yeah. So it's also a possibility that the ones that are transient, depending on when you sample the patient, whether they're actively infected or not, you might not catch them because they might have resolved by that time. So you end up getting the latent viruses.
Yeah, that's right. And I think that's, a really good point around any of the microbiome studies is that there's a big sampling bias based on sort of the timing of the sampling as well as the location. And I'd love to see a lot more longitudinal, high resolution longitudinal studies. But I think one of the big challenges, of course, is funding.
Yes, of course.
Yeah.
They tend to be more expensive, unfortunately.
Oh, yes. Yes, yes, yes.
All right. If that's okay with you, Stephen, we'll go ahead and ask Kevin's question regarding the protective commensal network. For Seweryn, do you see similar differences in nasopharyngeal colonization loads, given that there's a reduction in the bacterial load in the middle ear fluid?
Yeah, so for that particular study, we didn't do any sampling of the middle ear fluid. It was just nasopharynx. So that data is from the nasopharynx itself. These were children that were recruited, their ages 4 to 6. And most of them had a healthy tympanic membrane, but they did have a history of recurrent otitis media. Only a portion of them had either perforation or OME. So we didn't really get much of a chance to actually sample the middle ear fluid.
Yeah, is that okay, Kevin? You can also unmute if you want to.
No, that's good. Thanks, Seweryn. And I actually worded that backwards. Yeah, that was. I guess what I'm curious about, and I guess this maybe leads to whether you can find those protective organisms in the middle ear, because obviously once the otopathogens, if you will, get up to the middle ear and they establish infection, at what point could those be protective? It's an interesting concept, and I really like it. I was just curious if those are thought to be used as a probiotic effect. Maybe not necessarily that's the right word, but at that point in the middle ear itself.
Yeah, no, I appreciate the issue with the kind of the term probiotic, because that's a bit of I think a loaded term with a lot of different meanings behind it. But I think the protective element of it comes as a preventative. So it prevents the progression of those otopathogens into the middle ear. Once they're in there, I don't think the commensal bacteria really play as big of a role in terms of, limiting the infection itself. So I think the importance is the timing of it. They have to be there to prevent the overgrowth, the kind of the blooming, I guess, of the otopathogens. So they just keep them in check. So that's the general idea. Same thing with using some of these organisms as probiotics. There's been the ideas have been tossed around about using it at a very early age at these high-risk populations. So almost like, given to them like on drops on the breast of a breastfeeding mother to try and seed these healthy bacteria first, so that they can get in and establish themselves before the otopathogens get introduced into the nasal cavity.
It's quite striking because at least from first encounter of otitis media versus even a secondary, and you're just not finding them there. Is that correct? It's just, or their load is significantly reduced, these protective organisms.
Yeah, I think there's still a lot of work to be done, because certainly we were finding Delosi granulum in the children with history of otitis media, or also just with existing otitis media at the time of sampling, but they just weren't correlated with Corynebacterium.
Yeah. Okay.
So,
right.
so it has to be that interaction, that sort of solidified interaction between the two, that forms that protective effect. How that's happening, I don't think that's really been elucidated yet. Even like, with Catherine Lemon's work, they really didn't figure out what that particular mechanism was.
Right.
And it could be, sequestration of nutrients. It could be direct competition for space, or some sort of bactericidal effect. We just don't know. Yeah. So just on the back of that, some of Leanne Kirkham's work in Western Australia with Haemophilus hemolyticus she's done some nice work with the mouse model showing, and she's developing a probiotic spray using that strain that showed that it prevents in the mouse model the development of otitis media, but also there seems to be a immune modulatory effect, because they've also shown that it reduces the viral loads and overall clinical outcomes of influenza virus infections. So that, also holds a promise of, if you can use this probiotic to help prevent otitis media, it might have secondary benefits from reducing respiratory viral infections, at least temporarily, which then may in turn also then reduce the respiratory viral infection impacts on the perturbing the respiratory microbiome, and consequently, maybe that's the mechanism, again, of protecting against the development of OM, is that it reduces the impact of the respiratory viral infections, or both. Who knows, right? Clearly, we need more money to look at all this stuff.
All right. If it's okay, we'll move on to the next question for Diego from Brianna. You want to ask your question, Brianna? So you can unmute.
Yeah, sure. I was really interested in the proteomics of your middle ear effusions, and I have an obvious bias for wanting to know how much heme is in everything, because obviously that probiotic that you mentioned in your talk is very reliant on how much heme is there. And it's really hard for us to do studies that appreciate the conditions that are happening in vivo. We use culture media and stuff without really knowing what's in all these juices and stuff. So I was just wondering if you've ever tested, other molecules like heme or arginine, things that we know influence, Haemophilus and its competition or cooperation with other bugs in the.
No, it's a great question, Brianna. What a great question. So as I saw your question come in the chat, I went back, and this data. We did proteomics on a limited number of samples, and we did it back in 2010. So an unbiased approach to find heme proteins was a question that I didn't know off the top of my head. So I'll just show you a summary. I'll show you briefly the data. And we haven't, to answer your question, compared this sort of global protein abundance in the limited sample set from chronic otitis media samples we did. But you do find this hemopexin protein. I don't know what that is. I don't know if that's associated with heme or heme-like proteins. And in fact, when you look at the list of the 30 most abundant proteins by proteomics in chronic otitis media with effusion, that's this slide. I don't know if you can see that. Hemopexin is one of the 30 most abundant proteins through proteomics. How that correlates with the microbiome, I don't know. I don't think we did microbiome on the same samples. I'm pretty sure we did not. And so I don't know, but it's really an intriguing question. We haven't looked at those proteins specifically, but it would be a really interesting question to ask if, there's a correlation between abundant nutrients and, bacterial genre profiles at least. So good question. I'm sure there's some correlation there though.
Yeah, thank you. That's really interesting. So hemopexin is a heme binding molecule in the body. So naturally, our body doesn't want free heme floating around. So good to know that's there, and I might include that in my next experiment.
Yeah.
Brianna, I can jump in a little bit here. We did some proteomics work in our chinchilla model of otitis media. The number one hit on that list were hemoglobin binding proteins from Haemophilus that we identified, but at least from the bacterial standpoint, suggesting that, and we showed this from the host, in this case chinchilla, that hemoglobin was number one component. Obviously, being a binder of heme becomes very important in that model system as well. So yeah, it's obviously a big, at least a predominant player in nutrient, and this idea of restricting nutrients to limit growth is a fascinating topic. Something we've found, which is kind of a fascinating side kind of a effect of this nutrient limitation is that we believe this starts to trigger some pathogenic profiles in Haemophilus, and their behavior they adapt quite quickly to limitations of nutrients as well. So that's always the challenge that we deal with. So we obviously try to figure out when they're using certain nutrients, but then the effect on the bacteria because of that seems to be pretty dramatic. So it's interesting work, and I too congratulate on your paper that recently came out. So nice work.
Oh, thank you.
We'll go to Tanya's question. So this is for Seweryn and maybe Diego. So Tanya is asking, should she still do adenoidectomy, given that it's not an established reservoir for pathogens in the middle ear?
I might defer to Diego on this one in terms of changing management practices for patients. But look, it's still the jury's out. Essentially, we're not sure one way or the other. I think that's what the summation of the literature right now, that's the status of it.
You know, that's such a
May I ask a comment? I like take-home messages. Tomorrow I'm going to the hospital, to the OR, and do adenoidectomy with inserting grommets. Should I do it, the adenoidectomy, or not? Should I change my surgical practice according to the nasopharynx, the adenoid being a reservoir, or not? What do you say about Diego?
Hey, Tanya, I wouldn't change based necessarily on the fact that the microbiomes don't correlate. I do think there are certain clinical benefits of adenoidectomy in otitis-prone children. And if you look at the guidelines, especially older children, they seem to get long-term benefit according to mostly retrospective studies with adenoidectomy. I myself have become more selective with adenoidectomy for children that have otitis. I tend to perform it on children that have clear signs and symptoms related to adenoid hypertrophy, chronic sinusitis, or nasal congestion. Especially in younger children, I don't use adenoidectomy as a primary treatment of otitis media in younger children. And I think the concept that I have heard at some meetings that all otitis media is due to adenoid disease. I've heard people say that at conferences. I don't think that's true. I don't think all otitis media is due to adenoid disease. And I don't think adenoidectomy is a primary treatment of otitis media. That's just my opinion. But I think the reservoir theory is a question when you profile the microbiome. There's a question as to whether it's a direct causative or source of the bacteria. There's a relationship, I'm sure as you know very well, Tanya, and could teach us all, there is a relationship between the nasopharynx and the ear. There's a mechanical relationship. I'm sure there's some form of shared host immunity relationship. I'm sure there's some relationship with pathogens too. But I don't think it's the sole source, nor should it be the sole treatment source. And that's just my approach.
Thank you.
Everybody will give you a different answer, Tanya. That's the challenge. The guidelines help us a little bit, but even the guidelines are not perfect. So
if I may add 2 points. So the first point, I was looking for other control studies, and there was one in which they sampled the entrance to the eustachian tube, also swabbing a little bit of the adenoid. And of course, since you do it through the oropharynx, then it's also seeded by bacteria from the oropharynx and the buccal or oral cavity. So that's also a sampling issue. If your bacteria is contaminated on the way to sampling, then of course you will have a very different niche when you start profiling it. So that's one. The second is there are so many other things that we touched upon today. I'm kind of closing in a way, and I'm segueing. We touched upon potential new therapies, which is really exciting for probiotics. We also talked about microbiome profiles in indigenous populations, especially in Australia. Although we do have some webinars already set for the next 6 months, that would be acute otitis media and immune profiles, medical treatment, and biofilms. There are other webinars scheduled tackling these other interesting topics, so please just watch out for those announcements and we'll try to form them as we go along. Again, this is just the first webinar for the next 5 years, so we're excited to just have different voices and different perspectives discussed in these webinars, so then we can also encourage each other to continue with research.
Thank you
Right.
so much.
Thanks, Anya. If there are no more questions, for the rest of the audience, thank you so much for staying with us during this scientific session. Rita Lee is now here. We will start with the career advancement seminar. For today, we will be discussing collaborative leadership. You're free to stay if you want to participate. There's going to be some audience participation. But then again, if you have to go somewhere else, this is also recorded, so you can also watch it later. We'll give a few minutes for some people if they're logging out. Thanks again. All right, Rita, floor is yours.
All righty. So if everybody who is on can grab a sheet of paper and a writing implement, this is going to be active learning and not just a lecture. So let me see if I can get my stuff to work. Okay. Are you seeing my slide deck or my speaker's notes? You're good? Okay, thank you.
For me, I see the speaker.
The speaker
version.
notes?
Yes.
Same.
How do I shift that? I don't actually, is that better? Okay, now I lost you all.
You were at the right location. You just have to swap it, not duplicate it.
Okay, let's see. This is such a pain. Maybe this one. Does that work?
Yes.
Okay, now I can't see any of you. All right, well, I will keep talking.
You can't have everything, Rita.
I know I can't have everything. Anyway, welcome to all of you. Thank you. Hopefully it'll actually let me advance my slides here. So today we're going to be talking about leadership. Our goals for this afternoon are to compare and contrast different leadership styles, describe the process of collaborative leadership, and then analyze the strengths and challenges of collaborative leadership. So we're going to start with an activity. So here is where you are going to need your paper and pencil. And I think with this group, I think it might allow us to do breakout rooms now that, no, it won't. Okay, that's fine. So you are the member of a space crew originally scheduled to rendezvous with the mothership on the lighted surface of the moon. However, due to mechanical difficulties, your ship was forced to land at a spot some 200 miles from the rendezvous point. During re-entry and landing, much of the equipment aboard was damaged, and since survival depends on reaching the mothership, the most critical items available must be chosen for the 200-mile trip. So I'm going to show you a list of the 15 items that are left intact and undamaged after landing. Okay. Your task is to rank order them in terms of their importance for your crew and allowing them to reach the rendezvous point.
Rita,
Okay.
I
Yep.
can do breakout rooms for you. How many do you want?
I think 2.
All right.
But
I'm gonna
not quite yet. Not
Not
quite
quite
yet.
yet. Okay,
Yep.
just give me the go signal.
Will do. So on your sheet of paper, what I want you to do is, you don't have to write the whole thing, but just know what you're ranking. So you have one box of matches. You have food concentrate, a 50-foot nylon rope, parachute silk, a portable heating unit, 2 .45 caliber pistols, 1 case of dehydrated milk, 2 100-pound tanks of oxygen, a stellar map, a self-inflating life raft, a magnetic compass, 5 gallons of water, signal flares, first aid kit, including injection needle, and a solar-powered FM receiver transmitter. So what I want you to do is take about, I know it's fast, but take about 5 minutes, if you can, and rank these on your own in order of importance. So if it's the most important thing, it's going to be number 1. If it is the least important thing, you're going to rank it number 15. Okay, does that make sense? Okay, so I'll leave this up and you're going to do this on your own piece of paper.
Stephen has a question.
Okay, go ahead.
Hi, are we anticipating any source of danger?
You are on the moon, so your danger is dying on the moon.
Excellent.
Okay, but there are not aliens or Martians or Moonians or anything like that. All right. Awesome. All right, Reggie. So in a minute. So now what we're going to do is break you into 2 teams. Okay. And what I want you to do in your team is we will give you about 10, 15 minutes, maybe, well, let's see, it's 5:45. Let's say about 10 minutes to work as a team to come up with your team rank. Okay, and hopefully you all will have the opportunity to come to an agreement about what that's going to be, and then we'll have you come back and let us know what your team rank is. Okay. So Reggie, whenever you're ready, you can send them to their breakout rooms.
All right. This will be done randomly. See you in a bit.
Yes. Thanks. Were you able to make a plan, group 2?
No, we're still talking.
How far in your rank list did you get?
We
do have a rank list.
Yeah, we kind of discussed everything.
Yeah.
Okay. All right. So I'm sure you are all wondering what the experts have to say about what the quote unquote right answer is. So on your piece of paper, what I want you to do is draw another column that has the NASA astronaut ranking list. Okay. And then I'll show you how to calculate your scores and see how you did. All right. So this is the rank that comes from NASA. Again, there might be some conversation and debate about this. But your box of matches ranked number 15. Okay, so I'll start with number 1. Should have been 2 tanks of oxygen. It is the most pressing survival need because obviously without oxygen, you are dead. And generally weight is not a factor since gravity is only 1/6 of the Earth. So it really only weighs like 17 pounds. Okay. Number 2 should be 5 gallons of water because you're going to lose a lot of liquid on the light side and you do not want to get dehydrated and become, freeze-dried ice cream. Okay. Number 3 was the stellar map. That's how they navigate. And apparently the star patterns appear identical on the moon and the Earth. I did not know that. Number 4 was food concentrate, so you could have energy to keep that 200-mile walk. Number 5 was that solar-powered FM receiver transmitter, so you can communicate with the mothership Okay, it does require line of sight transmission and can only be used over short ranges. Then number 6 was the nylon rope, which was useful in scaling cliffs and tying injured people together. Number 7 was the first aid kit. Okay, apparently these needles fit like a special little opening in the NASA spacesuit. Number 8 was parachute silk to protect you from the sun's rays. Number 9 was that self-inflating life raft. And what they're saying is that carbon dioxide bottle in the raft can be used for propulsion. So I guess you just, and it like shoots you that way. Cool stuff, right? I didn't know these things. Number 10 is the signal flares. And so when you see the mother ship, you can use that as a distress signal. And then number 11 was the pistols, again, for a means of self-propulsion. Number 12 was the milk. It's basically a duplication of the food concentrate and doesn't give you as much nutrition. Then the heating unit, it's not needed, I guess, unless you're on the dark side of the moon. Then number 14 is the magnetic compass, because there's no magnetic field on the moon. It's not polarized. So that compass is worthless. And then, of course, the matches, because there's no oxygen. So what are you going to burn?
Would the pistols shoot without oxygen?
Apparently so. I have personally not been to NASA. You know, I've never been on the moon. So
Our discussion was about the signal flares as well, if they will work.
I looked that up, and they do.
Yeah, I'm sure they have special chemical ones. Like, I think it's some, I don't know. Again, the whole point is that none of us are experts in this.
I actually looked it up, and the University of Illinois detailed that they have special chemical flares to work underwater and ones that can work on the moon.
Okay. Right. So I'm guessing there's something about the pistols that don't necessarily need like oxygen for a combustion thing. I don't know. Okay, so now the next step is to see how your team did and how did you do. So what you're going to do is, does everybody remember absolute value? Right, so we're going to look at the difference between the NASA rank score and both your individual score as well as your team score. Right, so if you scored matches as a 15, you would get a score of 0. Okay, if you scored matches as number 1, right, then that would be a 14. Does that make sense? Okay, and so what I want you to do is now tally up what your score is and also tally up your team's score. I'll give you a couple minutes to do that. And then when you're done with that, I want somebody on your teams to tell me how you did.
So just to clarify, if we have the right one, so if we said oxygen, that is a 0?
Correct.
All
right. Yep. But if you said, like, for solar powered receiver transmitter, if you said it was 1, then your score would be a 4. If you said it was 15, your score would be a 10.
I'm confused.
Okay.
I don't know. I don't know how to score.
Okay. So you're looking at the difference in so start with your numbers.
Okay.
Okay. And then compare them to NASA's numbers.
Oh, okay. I got it. Okay.
And then
Thanks.
whatever that number is difference, just don't put negatives. It's going to be whatever the number difference is will be a positive number.
Okay. Thanks.
Uh-huh.
I'm probably one of the first to die. I got a 55.
That's, probably not a rocket scientist. So you're going to do that for your numbers, and then you're going to do the same thing for your team numbers. Obviously, I mean, everybody on the team should have had the same scores since you were a team. So only one person needs to be fast enough to do that math. Oh, thank you.
Yeah, I was, I guess, lazy or smart, I guess, depending on how you look at it, and I used Excel instead. So
Okay.
you have to stretch the boxes on your numbers.
So you already did the math, or you had Excel do your math. That's awesome. So Guillermo, how did your team do?
So we did pretty well on the top 5 items, and generally well on the least important items, but we never really ranked anything past, I'd say, like 7. So I can't really tell you the official score. But I think generally we were pretty close. We were really trying to discuss like how to get there. I guess the solution was to walk. And I guess anyone from the group can speak up, but I know on Earth anyway, 10 miles a day is about the maximum you can walk. So maybe on the moon you can jump or something and you get way farther. I don't know. Or you shoot yourself. I didn't expect that to be an answer. You shoot down and launch yourself away. So yeah, a lot of clever solutions. Or someone mentioned using the raft as a skid on the surface. So a lot of creative ideas.
Cool.
I would say our team got around a 25 based on.
Okay. What about the other team? And you were team 2, right?
That's right.
Okay. Team 1, how did you do? We're not done yet.
I definitely would die. Because I'm 53.
The official scoring, they say if you're 25 or less, you did pretty well. You would probably survive. If you're somewhere between 25 and 50, you'd be pushing it above 50. They, suggest what they call earthbound logic. Probably would have flunked out.
I told you I'll die.
Team 2 is pushing it at 38, so we are 50-50 surviving.
Okay.
Yeah. The heating unit really killed us. That was plus 11 for us. We ranked that one pretty high because we thought it got freezing cold at night. So
yeah. All right. Is everybody done calculating, or there's still some people crunching numbers?
Well, does the scenario say if we landed on the dark side of the moon or not? I mean, who knows if we need the heating unit?
Yep. Right. So it said that you are on the lighted surface of the moon.
Disappointing.
Okay. Anyway, obviously the whole point is the idea of how do we interact with each other and come to a team conversation and team decisions when none of us maybe somebody here is an astronaut, but most of these are designed so that the vast majority of people are not the experts in this content area. So here's a list of questions that we're going to talk about. Did anybody do better as a group or individual? Who did better as a group versus the individual?
I was better as a group.
yeah, I was as well.
Yes.
Totally.
By 1.
By 1. Okay. Anybody do better as the individual? Okay.
I actually did better as an individual because I had the heating unit very low.
Okay.
But they convinced me that heating was important, so I believed them. But
okay. Yeah, so most of the time, the group, right, the difference in perspective, that dialogue, and I know we kind of rushed it a little bit, is going to make for better decision making than the individual, right? Because it's theoretically shared knowledge. But sometimes it can go a bit awry. So what were some of those factors that you think led to the group doing better? Except maybe Landon. Landon probably would have been better without any of us. No.
I believe different point of views
Mm-hmm.
can make us think differently about a subject. So I guess this is the most important influence when we talk about different people talking about the same subject.
Guillermo?
Yeah, I think there was like shared knowledge. Some of us knew some things about, you know, some of the items, or creative use, I guess creativity too, shared creativity for the items.
Mm-hmm. Okay. What else? Was there anything around group dynamics that potentially positively or negatively contributed to higher survival? And group dynamics is like how you interact with each other. Are we actually listening to everybody? Are we not listening to everybody? How is everybody's input considered?
I think we started to have at one point, you know, fun, and, you know, started to come up with like more or less crazy ideas, you know, and I don't think I would have gone there if we hadn't been in a group and like would have sort of brainstormed on, oh, and we could have done this or that, or, you know, I think it allowed like more creativity.
Mm-hmm. Okay. Jason, I saw you unmuted for a sec.
I felt because it was a pretty short amount of time. And so I think it's, you know, the group dynamic during that short amount of time was very good. I mean, everyone was like very respectful, and, you know, with the Zoom things, like what, you know, when we were starting to talk at the same time, someone would like let the other person talk kind of thing. But then I wonder if in a real situation, like it was like Survivor, you know, then the group situation might be really different. But I think in the short amount of time, everyone was like very respectful and let everybody else talk. And
Mm-hmm. Are your teams normally like that?
Well, that's why I say I think normally they start off really nice, and then stuff happens. So
Yeah, I feel like the other day we had a kid code and everybody was not like talking like this. And it was very not like this, and it was different. And I can definitely I agree with kind of what Jason's saying. Like different situations, people definitely would react differently.
Mm-hmm. Okay. Yeah, and what the other thing we want you to think about, right? Obviously, this was a fun activity to kind of bring out some of these. And as you all pointed out, like as your group kind of forms and changes, sometimes those dynamics are going to change. But what we want you to think about is like, you know, how did you actually embrace in a diversity of opinions? Were there, and I'm not saying arguments, discussions of like heat lamp or no heat lamp? Right, Landon, and I'd love to hear your thoughts about how that conversation or discussion about like the diversity of opinions around the heat lamp, how did that play out for you all?
Honestly, I just kind of took the back seat. They were all like, we should prioritize like the primary needs first, so like food, water, and then like shelter was kind of just heat. They said that heat was the next like big thing. So I just kind of did like a group think and just went with them. I was like, okay, they probably know that the moon's colder than I do, and I just went with their opinion and didn't really question it or give a differing opinion at all.
Mm-hmm. So the interesting, Landon, so I was in the same group. So I had heating as number 13 because I was like, well, aren't we in a spacesuit and shouldn't that protect us? So why would I schlep a heating unit with me? That was like my, you know, like my personal thing. And then everyone was like, oh, we need heat. The moon is cold. And I'm like, well, I have no clue about the moon, you know, so it must be cold there. And so that's why I didn't, as you, Landon, didn't say anything, you know, because I'm like, well, that's logical.
Okay, so not, you both had it ranked pretty low, but neither of you actually mentioned that.
Yeah, I had it at 14, and I didn't think it was a priority at all because I just felt like it was a lot of extra weight, but I didn't challenge it.
Okay. Interesting. Right. And that is something to think about, right? Like as we are leaders of our teams of, right, this wasn't actually an active conflict, right? Because none of you actually said like what your thoughts were out there. And so just thinking about that as we think about like group dynamics, you know, what potentially could we have done a little bit better to work as a team? Maybe some of us would have survived instead of just perishing on the moon out there because we're schlepping a heat unit unnecessarily or something else.
I mean, I that's probably my fault. I take responsibility of that. I had ranked it pretty high, and I think I was like, when we were talking about our top rank list, I was like, isn't space cold? And then I think someone else was like, yeah, space is cold. And then
Yeah, and that made total sense to me. And therefore I was like, oh, yeah, that makes sense. I agree now with putting it up higher. So
I just decided to just Google and look it up. And in the dark side of the moon, it's very cold. On the light side of the moon, it's extremely hot. So that would have probably cued the logic to be like, wait a minute, there has to be something more to regulate this. Because why bring a heating unit if during the daytime it's, hang on, let me check, because I have it up. If it is during the daytime 121 degrees centigrade and negative 246 when it's dark. That might have, knowing that might have probably helped everyone.
Yeah, right. Obviously, the whole point is like not to Google everything and know what the answers is, but right, like we make assumptions, right? Because none of us know exactly what the right answer is, and we're not supposed to Google it during this activity, right? And it's a lot about like, how are we allowing people to contribute their ideas? Are we withholding our ideas from a group? Are there ways that we can actually work better together? As a group, I'm going to share my screen here, right? So number one is we definitely, it helps us to know kind of what Jason was alluding to, that all groups have their life cycle, okay? And this is what we call the Bruce Tuckman model. And basically when groups are first forming, right, you got into your groups, you didn't really have time, you were just going to be nice to each other. Okay. And for a new group where nobody knows anybody at all, what they're going to do is really talk about like, do I fit in with this group? Most people are going to be talking about really like, you know, informal, casual, light things like, oh, let's talk about the weather, or oh my gosh, the air quality, right? You don't meet somebody for the first time and immediately say, so who are you voting for? What are your politics? What do you think about abortion rights and, you know, gun violence or whatever it is, right? Like most people don't do that. Okay, it's going to be a lot more superficial. Then when you get into a group a little bit more, most groups have to go through this phase. It's called the adolescence of a group. And this is where you're storming. Okay, so people are going to be like, well, how am I different than you? How do we organize ourselves? Sometimes little cliques start to form and people start to push back against you as a leader. And this is normal. And this is what we want everybody to understand, is some of this is normal, and how you manage it as a leader can impact, right? You have to allow some of these conflicts to occur. If you can get through that storming phase, then you get into a norming phase, which is step 3. And that's saying like, what are your strengths? What do you know? Let's figure out a way to work together. Okay. Ideal world, you get into performing, which is really highly effective teams where you collaborate or compete in a friendly manner. You have an effective structure. Everybody knows kind of what their roles are for the most part, right? We can say Reggie's expertise is this, and Brianna knows this, and Jason, right? And Guillermo's phenomenal at Excel spreadsheets. And so you can say, we got to do this. Well, at least you did it, right? I don't know. You know how to do an equation. I don't know how to do a formula in Excel. So you can then say, let's divide and conquer and let's leverage our strengths. to be able to get this done. Okay. And then when a group is over, it's called adjourning. Okay. So it's important to know that as you kind of think about leadership, especially collaborative leadership, because your goal is to encourage some activity so people move quickly through forming, get through the storming phase, and ideally end up in norming. I will tell you probably something like 80% of groups get stuck in that storming phase because we're not willing to share. Right. We're withholding our information. We're not saying, oh, I put heating lamp 13. I guess, okay, I'm just going to go along with whatever you went. Okay. Or like, you know better and you're like, oh, well, I'm going to let them suffer. Okay. There's also multiple different leadership styles. Okay. None of them are necessarily the one right way of being. Okay, so when we think about leadership styles, there's the autocratic authoritarian, and that is the decision, right? Like me as the speaker, I'm telling you, do this, do that. Okay, it's not necessarily like a dictator, but it can get to that depending on how bad your person is, right? So the pros are, as a leader, is you get control, you have discipline, it's relatively quick. Okay, the cons are, people lack freedom. There can be a lack of trust. There's no professional development, and they're overly dependent on the leader. Okay. And I'll be curious to group one who came out early as we go through this. Which one were you? Okay. The next one is democratic or participative, where the decisions are based on input. Everybody, you consult with everybody, and there's engagement in that way. The pros is that people can evaluate their own performance. Employees can actually establish their own goals and grow on the job. And you're getting in useful input from everybody. The cons are, as a leader, sometimes they tend to blame everybody else, right? Well, the group wanted this, so it's not my fault things failed. Okay. Sometimes quiet team members may not share important suggestions or feedback unless they're prompted or asked. And then other employees maybe just don't want to be involved. Okay, laissez-faire leadership style is basically the decisions come from the employees and you're basically kind of absent or useless as a leader. So it can be helpful when creative ideas are important because you're just kind of like hands off. Okay, and it can be highly motivational for some people who are really self-regulated and can get their work. Okay, the challenge is that it may not always work. It's really good for teams with experts or specialists, but not people who need a little bit more guidance. Okay, it does rely on good teamwork. Okay, transformational leadership is pushing people to think and work in a new way, requires strong motivation. Employees tend to grow with the group or the company. Okay, and there can sometimes be a bond between the leader and the employees. However, it only works for employees who actually want to be part of that process. Sometimes can result in favoritism because you're leaning on like, oh, what's, Antonio's new ideas or Helen's new ideas. And then transactional is rewarding people for what they do. I pay you, you show up, you do the job. Okay, rewarded followers tend to become more motivated. There's a clear structure. If you do A, I give you B. Okay. However, it can sometimes constrain establishing trust. The follower sometimes may not accept corrective actions of the leader, and it can breed like that what's in it for me kind of an attitude. It can actually make sometimes things worse of like, well, why should I do that? Because I'm not getting anything anymore. And then finally, collaborative leadership is that leadership shown by the group that is, again, acting collaboratively to solve agreed upon issue. Okay, it's multiple interested parties that have different perspectives. It can lead to really innovative solutions. People working in alignment can actually get more done. And maybe that is what Team 1 did because they were so collaborative, they came out quickly. I don't know. And you need it to solve really complex problems when there isn't always a clear answer or a solution. The cons are that it requires time and building trust. And no one is actually in charge or obligated to be responsible. So if nobody wanted to, quote unquote, play in the sandbox, you don't have to. So I'm curious with all of these, if your group tended to follow one of these or a different one, or did anybody actually take leadership of these teams?
I would say Matt definitely directed our group to get things started.
Mm-hmm.
Yeah, I just started talking.
Okay.
But then yeah, it was just I think then everybody was just kind of chipping in. I was like, what'd you guys put for 4? What'd you guys put for 2? And then we just kind of went from there.
Mm-hmm. Okay. So maybe a little bit more like
Agreed.
democratic.
Yeah.
Yeah, I'd say definitely.
Okay.
Except for Landon, who didn't participate and add in his opinion on the heater, which would have really helped us.
Well, you know, you're now assigning blame. No, I'm just kidding.
Was Landon in your group as well?
No, I was only in group 2. I don't
Yeah.
know how I.
Oh,
came into
sorry.
this discussion.
I just know you, so I assumed you were in group 2. Our group, my bad.
See, this is why we brought the guns for the other group. We knew this would happen.
All right. Well, in order to do collaborative leadership, right, part of it is you actually have to come to a shared understanding of what the problem is you're trying to solve, right? Because we could say, you know, we're trying to solve like, you know, cure otitis media, but like, right, there's so many different angles of doing it. Is it taking out the adenoids? Sorry, I just listening into the last part of that conversation. Is it antibiotics? Is it this? Is it that? Is it heme? I did not understand any of the science behind it, but cool stuff. Right? Like, where is the actual problem? Okay. Then you have to identify who are the key interested parties in this work. Okay. It's not just the researchers or the scientific team, but we also have to think about like who might be the end users or the patients, particularly in this. Yeah, Landon is a solo hiker, so he's going to survive. Yeah. Right? Who's involved? Who might be people who might be potentially like paying for the research, right? Then there's the people who might be doing the research from different angles, and then the people who are going to benefit potentially from the research, and they're all going to have different perspectives of what is important in this space. Okay. You have to then assess the extent of interested party agreement. Like, how much are we actually agreeing that this is the thing that we're going to work on for now? Okay. Or are people going like, sure, fine. Right. And then Landon and Sonia are like, not really. We're going to go chuck our heaters over here. Okay. Because we're not schlepping those things, but we're going to go do our own thing. And then you don't actually have a collaboration anymore. You want to evaluate the group's capacity for change and to actually implement something that is actually different than what the status quo is, and identify where the problem or issue can most effectively addressed. Okay, thinking about this like as a team. And then finally, the key skills that are required for collaborative leadership is number 1 is creating the opportunity for everybody to participate in decision making. Okay, so there could have been, and I'm not saying you did it wrong, because there's no right or wrong to this, right? But there are strategies where you can effectively ask everybody for their input. You can create space for people who may be quieter or not necessarily willing to share, or maybe even afraid to share because they're worried they're going to have the wrong answer. You can also have like voting versus consensus decision-making strategies that allows everybody's input. Okay. There's a great website called Liberating Structures that actually has different tools and strategies like open space and other opportunities for get everybody's input on a particular topic or a question. You have to have effective information exchange. So multiple avenues for communication. Some of us are auditory learners. Some of us are visual. I'm a tactile learner. Like, I do better if I take notes, right? Sometimes email, social media, et cetera, et cetera, et cetera. And thinking about how are we going to communicate with each other? Are we going to do meetings? Are we going to do this through email versus phone? Thinking about inclusive access to technology. Think about how are we going to manage conflict? If we have a disagreement about the heaters or oxygen or the guns, et cetera, how are we managing that? Okay. Are we going to be competitive? Are we going to just be avoidant conflict style like Landon and Sonia had, where we're just not even going to say anything about the heaters or where we ranked it? Okay. We have to have shared vision and values, and you have to be able to have a conversation around that. You want people to get to that point where they can commit to that common goal, cause, or issue or problem that you're actually going to working on. You also want to think actively about how are we making decisions? What is that structure? What is that process? Are we going to be unanimous? Is it consensus? Is it majority? Is it voting? Is it going to be anonymous, etc.? And I'm curious, in your groups, how did you actually get to those decisions? Right? Sometimes whoever has the pencil and writes down the numbers is the one who gets to control everything. Okay. But there might be dissent in that. Okay. Getting to the point of unanimous does take some time. Okay. Sometimes you end up consensus is easier. All right. And then of course you have to have mutual respect and or at least tolerance for differences of opinion. Okay. So that is collaborative leadership in a nutshell, hopefully with a fun activity to kind of get you thinking about all of the different aspects that actually create a truly collaborative environment. And so I will stop there and see if there's any questions, comments, or thoughts. And hopefully you had fun at the same time.
Thank you so much, Rita. Just reflecting on my own experiences, I think I've gone through all those different styles of leadership depending on who I'm with. And I've experienced both the pros and the cons. So I think for those who stayed, it's a huge advantage that Rita is sharing these nuggets of wisdom with you. I wish I had them when I was younger. It would have helped me. And I would like to have a copy of your slides even just to reflect on them once in a while. And it might help me or guide me on what to do sometimes when I have something in my mind. Yeah. So are there any questions or comments from the group? Anything about collaborative leadership for Rita, the previous session, or any activities in the coming weeks and months? So I suppose we're closing. Nobody wants to chime in.
Okay, sounds good.
All right. Please do answer the surveys for everyone. Your feedback is really important for us to improve. And thank you so much, Seweryn, for your talk. It was great. It was really, really great. It helps us set up all our ideas. All right. Thank you, everyone.
Thank you all.
We'll see you soon again.
Take care.
Bye.